Good morning, everyone, and thank you for the opportunity to present my work. Hemorrhagic shock is the leading cause of death on the battlefield, contributing to up to 50% of traumatic injury deaths. The kidneys are one of the first organs to succumb to ischemia due to hypoperfusion and to become injured during resuscitation due to damage to renal vessels. These events lead to immediate kidney injury and subsequently renal dysfunction long after recovery. This highlights the fact that renal injury at the time of hemorrhage and resuscitation can lead to increased risk of renal-related disease later in life. The use of vasopressors during hemorrhagic shock is controversial, with some evidence showing their use can lead to further tissue ischemia, acidosis, and vasomotor collapse, while other lines of evidence demonstrate increased organ perfusion by venoconstriction and increased cardiac contractility. A paper published in 2016 actually shows that vasopressin is renal protective and can attenuate kidney injury during hemorrhagic shock and resuscitation. This suggests that vasopressor administration during hemorrhagic shock resuscitation may lead to molecular and physiological changes in renal vessels that can have an acute and chronic impact. Thus, in this study, my objective is to elucidate novel molecular mechanisms that underlie hemorrhagic shock-induced renal vessel injury and understand how vasopressor administration can alter this. This can help in the development of treatments and management strategies and in the elucidation of biomarkers of injury. An unexplained mechanism of hemorrhagic shock and resuscitation-induced renal vessel dysfunction is epigenetic enzymes. Epigenetic enzymes are responsible for adding or removing molecules from your histones and DNA that cause your genes to turn on or off. Changes caused by epigenetic enzymes largely explains why identical twins are not truly identical and can develop different disorders, or why one twin would become a family practice physician and the other a general surgery physician, even though they share the same sets of enzymes. Given that epigenetic marks have been discovered to be reliable biomarkers of acute kidney injury and vascular disease, and that they can remain stable for years, I propose that the alteration in epigenetic enzymes underlie the changes in renal function caused by hemorrhagic shock and vasopressor administration. My hypothesis is that epigenetic enzymes in renal vessels are altered in a vasopressor-dependent manner in a pig model of hemorrhagic shock. Our experimental setup included 22 pigs, which were put under general anesthesia and catheterized. 6 pigs did not undergo hemorrhage, while the rest were hemorrhaged to a target pressure of 30 millimeters mercury, or approximately a 45% blood volume loss. After 60 minutes of hemorrhage, pigs were resuscitated with normal saline to a target pressure of 60 millimeters mercury. After 60 minutes of saline resuscitation, pigs were given additional saline or vasopressin or norepinephrine. Vasopressors were discontinued after 60 minutes, and the pigs were observed for a total of 180 minutes after that, after which they were euthanized and vessels were collected for real-time quantitative reverse transcription PCR later on. And then interestingly, when I probed the expression of 84 different epigenetic enzymes in the renal arteries, I found many differences, depending on the type of resuscitation and vasopressor used, thus supporting my hypothesis. These graphs demonstrate all the enzymes in the PCR array that had a statistically significant change in the amount of gene expression, with fold change from the non-hemorrhage group on the y-axis. They highlight the variety and degree to which the enzymes were affected. Compared to the non-hemorrhage group, the normal saline group affected a smaller proportion of enzymes than the vasopressin or norepinephrine groups, and in general, the norepinephrine group elicited larger magnitude changes. Furthermore, the vasopressor groups upregulated expression of the same families of enzymes, to include histone deacetylases, the set domain containing methyltransferases, and demethylases, while specific to the norepinephrine group, enzymes within the DNA methyltransferase family were upregulated. And then this diagram, again, focuses in on the enzyme families shown in black boxes and demonstrates that while there was significant overlap in the upregulation of certain enzyme families amongst the two vasopressor groups, the norepinephrine group alone upregulated several enzymes within the DNA methyltransferase family. This suggests that the epigenetic changes are not random and seems to indicate a concerted response to regulate downstream expression of genes. I would also like to highlight that these epigenetic families have been shown to modulate vascular physiology in many different contexts as shown. And notably, the inhibition of HDACs have been shown to improve survival during hemorrhagic shock, making them potential therapeutic targets. While on the other hand, little is known about the role the other enzyme families may play in hemorrhagic shock and resuscitation, as we are the first to show their involvement. In addition to the dramatic changes in epigenetic enzymes, we also detected significant group differences in renal vascular resistance. After hemorrhage, additional saline only decreased renal vascular resistance by over 60%, and similarly, norepinephrine administration decreased renal vascular resistance by about 40%, while vasopressin, as you can see in orange, had a dramatic opposite effect on the renal vascular resistance. However, this effect was transient and disappeared after the vasopressor administration was discontinued. Overall, these data strongly suggest an association between epigenetic enzymes and the physiological consequences of vasopressor administration in the renal vessels and renal function. Until this study, little was known about the epigenetic enzymes in renal arteries and are significantly changed during hemorrhagic shock resuscitation and vasopressor administration. We are the first to demonstrate the dramatic effect of hemorrhagic shock and various resuscitation strategies on the renal artery expression of these 84 different epigenetic enzymes. This study strongly implicates vasopressor use in modulating renal function during shock and in affecting renal function long after recovery via epigenetic processes. These findings can serve to develop treatment therapies to aid in the recovery after hemorrhagic shock resuscitation, help identify biomarkers of injury severity, and lastly, to help expand research into the clinical applications of epigenetic enzymes. This concludes my presentation. Mahalo. I am happy to answer any questions at this time.